HPLC lab course, software ChemStation. This video is for internal use only, for example for teaching the learning content. Passing on or distribution in any form is prohibited and is reserved only for the owner of this video. First, the instrument software ChemStation will be opened. Load the measurement method, which is here parabene C18.m. Above, next to the green book, is the method information placed, where you can write in everything important about the method. Here, for example, IA lab course, the column's name Zorbax Eclipse XDB C18 and its dimensions of 150 mm in the length, of 4.6 mm in the inner diameter and 5 micrometer particle size of the stationary phase, and the flow rate of the pump with 1 milliliter per minute. Below the method information are all modules of the HPLC listed, which were explained before in part 1. The automatic sample system, the pump, the column oven, and the diode array detector. In the last part, the so-called data analysis and report options take place, which are not be used here. Now have a look at every module, what could be adjusted there. To do so, click on the automatic sample system. On Setup Injector, then another window will be opened, where it's possible to adjust the injection volume. Here 10 microliter. Then have a look at the module pump. Here is the pressure and the flow rate displayed directly. 132 bar and 1 milliliter per minute flow rate. When you click on Setup Pump, it's possible to see the pump parameter more in detail. Here takes the adjustment of the flow rate and the composition of the mobile phase place. Here is a possibility to change from an isocratic mode into a gradient mode. In the isocratic mode, you don't change the composition of the mobile phase during the whole run. Showed left-hand sided. 35% water and 65% methanol. This is what we use for this run. In the gradient mode, shown right-hand sided, you can increase the elution power by increasing the proportion of methanol for a certain time. Here shown at 1 minute run time from 65% to 75% methanol. Now click on Setup Column Oven Thermostat. Here is the possibility to adjust the temperature of the oven, where the column takes place, and the control of the column switching wave, where it's possible to decide which column should be used for the measurement. Here is column 2 selected, that means the C18 column will be used. Column 1 is here the C8 column. At least have a look at the DAD parameters. When you click on Setup DAD Signals, you can adjust the optimum wavelengths for your analyte parabenes. Here 254 nanometer. But during the whole run, a wavelength scan from 190 to 400 nanometer will be performed, because only the UV lamp is switched on. When you switch on the VIS lamp, it's possible to scan from 400 to 800 nanometer too. All module parameters were adjusted to ideal values for the parabene method in a method development before. In the lower section, it's possible to open the so-called online plot. Here would be shown the online chromatogram during a run. displayed as the red signal at 254 nanometer, and the pressure of the mobile phase displayed as the blue signal in bar. Both signals were plotted against time in minutes. Measurement of a parabene standard solution with a C18 column in isocratic mode. You have two possibilities of measurement options. First, with a sequence table. If you want to measure more than one sample, it's possible to write a list with the information of sample names, the vial positions, and the measurement methods. The automatic sample system would process from top to bottom the list until the last sample is measured. The second option is the single injection. If you want to measure just one sample, you have to define the operator name, the data file path, the file name, the vial position, a sample name, and you have the possibility to write in a comment. The measurement method is here the one which is already loaded in ChemStation. At least start the measurement via Start button. The automatic sample system. Injection. The arm of the automatic sample system takes the sample vial from the position which were defined in the software before and brings it to the injection needle. It peers the septum from the sample vial and takes the injection volume which is defined in the measurement method, here 10 microliter. After injection, the arm brings the vial back to the defined vial position. The red vertical line in the online plot marks the injection time. Question: how many peaks do we expect in our parabene mix standard solution measurement? Of course, we get 4 peaks. The standard mixture of parabene contains 4 parabenes: methylparabene, ethylparabene, propylparabene and butyparabene. When the measurement method is ready, a report window will be opened automatically. Above the chromatogram are written the operator name, the vial position, the injection volume, the measurement method, the method information and the sample info. Direct under the sample info you can see the chromatogram, and afterwards a table with important values. Retention times in minutes, the area under the peaks in milliabsorbance unit seconds, the width at the half high of the peaks in minutes, and some other values which are less important here. Here you can see the report again as a one-pager, with important values marked in red. These are values which are important for your calculations in your lab report. But how do we know now which peak belongs to which analyte? Answer. What do we know? First, we know it's a standard solution. That means there should be no other peaks except the analytes. Second, we know our column. It's an RPC18 column, a non-polar column. That means non-polar analytes stay longer on the stationary phase than polar analytes. Third, we know the polarity of the analytes. The polarity decreases with each additional methyl group. This allows only one conclusion. The retention order can be now clearly defined in the shown way. The first peak should be methylparabene, the second ethylparabene, the third one propylparabene, and the fourth one butylparabene. Measurement of the sample solution by the C18 column in isocratic mode. Measurement as single injection, same procedure as before. Now again the question, how many peaks do we expect in our sample solution? Answer. What do we know? We know the ingredient list of the after sun lotion. It contains methylparabene and propylparabene. And we know that we add an internal standard solution which contains ethylparabene. That's the reason why we get 3 peaks. And again the question, which peak belongs to which analyte here? Since there may also be other analytes in the sample matrix that are UV active, it is not only possible to argue by polarity alone, but the retention times of the parabenes are known from the standard solution, so that an identification by comparing the retention times is possible. Since the matrix from the sample has an influence on the retention time, the retention times only match to 1 decimal place. However, this is sufficient for an identification. Here is a summary of the identification. As can be seen here, there's a peak at 1.538 minutes, which belongs to the matrix of the sample and cannot be assigned to any parabene. All other measurements were performed as described before. This was the last video of this HPLC lab course. Thank you for your attention.